This continues the practical part of the program. Here's the short version, maybe an illustration. Yesterday I was having lunch with Garrett and he asked me a pretty straightforward question. He said, "You think you'll ever get a handle on this healing stuff in your lifetime?" And I said, "No." And so I've been doing this for quite a bit of time, but I don't really have a handle on trying to understand it. It's my primary goal. But in the meantime, because it's probably not going to be finished in my lifetime, I'm now trying to develop practical applications. And so this talk is primarily about practical applications and sound systems. So I've done a lot of experiments. I'll roam up here for a while. I've done a lot of experiments. These are just in vivo experiments on mice. So I have a bunch of them in a whole variety of places. I have a bunch in a whole variety of places. I have an equal number of in vitro. I have an equal number of this, that, and the other. I'm going to be focused on the practical application. So this is primarily for the IRVA folks who aren't particularly familiar with my research. This is going to be a real short 2-minute summary. Healing happens. And I can demonstrate that it does not have a decline effect under certain conditions. We can get a very reliable phenomena from healing. I'm reasonably confident, and I'm probably wrong, but I'm reasonably confident that there's a dose response. And so we've done this is going to would be a separate talk. I could go into all sorts of dose permutations, but healing is not magical. Too many people think of this as magical thinking. It's conventional versus this. Healing conforms to law-like patterns. And so I'm reasonably confident that there's a dose response. I'm reasonably confident, and of course I always add the caveat, probably wrong, but I'm reasonably confident that healing is nonlinear. You don't have an accumulated effect that has a symptomatic correlate. Rather, healing converts to something along the lines of a phase transition. Nothing seems to be happening, nothing seems to be happening, nothing seems to be happening, and then there's a sudden dramatic transformation. I'll show you a little bit of that. I'm reasonably confident, probably wrong, that healing has no energy involved. I know that's heretical, and that would be a separate talk. I think it is rather information. I'm reasonably confident that there's a resonant bond between the healer and healee. It's not entanglement, it's a fluid connection. And there are patterns which increase the bond, and there are patterns which decrease the bond. I'm reasonably confident that healing has very little to do with conscious intention. I understand that that's heretical also. That it is not necessary for a person to get into a particular state of mind that produces healing as an outcome. Rather, we have the causal sequence reversed. Healing happens, and the person has the option of experiencing that healing or not. A sensitive person will experience it, a non-sensitive person won't experience it, but it has no difference in terms of sensitivity or what you experience in terms of the functional outcome of the healing. Separate talk. And I could go on, but I don't have that much time. So for today, the question is, can we capture healing, reproduce it? Does it store? Can we capture it and then deliver it through some mechanism? And so I'll give you an idea of the healing, and again, this is primarily for the IRVA folks. What I do, and the models that I work with, I take a standard off-the-shelf model that everybody knows about, that's been studied to death. You know the outcome, it's a reliable outcome, there are thousands of published studies on this stuff, and then I insert a single variable, which is my healing technique. So we know what's going to happen, what happens if you put this into the system. And so I've done this certainly with hundreds of people, but I've also done it with hundreds and hundreds of mice, and as well as other carriers of this. So this particular model that you're going to see is a very tight model. It's a mammary cancer. We have 100% death within 27 days. The standard deviation of death is 3 days. It's a very tight model, and most people doing research are trying to get the distribution to change to see if they could get, for example, a normal distribution to look a little bit more like a negative skew. 100% of the mice die in 27 days. They die nastily. It's a non-metastatic. It grows, and then the mouse dies of either the internal organs being crushed and/or malnutrition. It's ugly. We insert a variable. This is me at Brown, dressed funnily, and we use my healing technique that I developed. A whole bunch of you know about this. And the question is, what happens when you insert this healing technique into a model which has 100% death. And what we get is quite anomalous. The tumor grows. Healing does not conform to the wishes of the healer. It's not a psychokinetic effect. You can't direct this thing. Healing happens. The tumor grows. It looks like it's looking bad. The thing starts to slit. It starts to ulcerate. First time this happening, I'm panicking because I think everybody's dying and suffering. But if you can see the coat from where you're sitting, it's clear. If you can see the eyes are clear. This is a healthy mouse that's ugly. Soon after this, I'm not showing in this, the tumor will implode and the mice are cured. I'm going to show you a phase transition here just because it's very cool. This is a different mouse. This is a mouse 22 days subsequent to injection. You can see an ulceration, I hope, from where you sit. I'll show you the same mouse 6 days later. Just because that's really ridiculous, I'm going to do it again. If you're not pulling back the fur, there's no sign this mouse ever had cancer. Nothing's happening, nothing's happening, nothing's happening, they're cured. That's the pattern. Sudden phase transition. We can do this with people. We've done it with a few hundred people, although it's reasonably hard to get people into those little cages. So we tend to work with young people, small people, and ectomorphs. So here's the question. That's the phenomenon in a nutshell. How do we capture it? Can we capture it? Can we store it? And can we make it a scalable phenomenon that can be delivered? So we got two questions. How to capture it? Can we recognize when it's been captured? And can we deliver it? That's the plot. Healing seems to be able to be stored, and just looking ahead very quickly, it can be stored in both biological systems, meaning life. It can also be stored in stuff. The stuff that we've stored it in can be organic. The stuff we've stored it in can be inorganic. This probably teaches us something. I'm just not bright enough yet to figure out what it teaches us. So we can store it in life. We can store it in stuff. We can store it whether that stuff is organic or inorganic. So from biology, the mouse you saw, for example, that's going to cure, seems to be cured for life, and these mice which are cured are immune to cancer for the rest of their life, and we can re-inject them with cancer. They can't get cancer again. So we can take, for example, these are actually rats because I couldn't get good pictures of mice, but we have a rat over here that has been treated. It's gone through the ulceration and it's done all those things, and here we have a mouse that has, or it's a rat, that has cancer. It's got the tumor, all that kind of stuff. If we take stuff out of this, and it can be blood or cells, and we put it into this mouse, no human intervention in terms of healing, everybody's cured. That indicates biological storage of the healing information. What if we do it to stuff? Well, here's organic material. We have treated cell medium. This is the stuff in which cells are grown. We treat the cell medium, and then we put cells into the cell medium. You probably won't be able to read that. We grow cancer cells. We did this primarily with human leukemia. We're about to start a replication in Providence. But we put human leukemia in the treated cells, and the cells probably don't believe much. But we put the cells in the treated medium. After 1 week, nothing happens. But in the second week, you have a sudden explosion. The cancer cells grow at an approximate rate. Yeah, the average is about a 600% increase in cancer growth. Some people get upset with cancer growth. If you're treating cells in a well plate, they're your client. Cancer is your client. Cancer likes to reproduce. This is another theme. Healing is a response to the need of the healee. Speculation, if we take that and stick it in a mouse, I suspect it will collapse. Haven't done it yet. We can take treated water bottles. We do the healing technique on the water bottles. We feed that to mice we never meet. The mice are cured. And so we've got something is storing, I think, pretty obviously. Now the question is, how do we detect and how can we tell whether or not it's stored? Can we tell by gizmos? Can we tell by stuff? Can we tell by mice? Can we tell? How do you tell whether the thing is stored? So here's my team. A couple of these folks are in the audience now. This is John McMichael, who's hiding over there. Mike Turner's hiding up there. Dean Radin, who many of you know, is, I think in Petaluma, taking people's temperature. We don't know why he does this. He goes around taking people's temperature, so he's doing that. And Sarah Besum is our lab person in Providence. Hopefully she's working. I don't know if you give her Monday or Sunday off. So the question is, if we could store and deliver this, is the best solution biological or is the best solution through stuff? If we go biological, we got an FDA. If we go through stuff, I could maybe, perhaps, hope we can do an end run around the FDA. So a physical solution is more scalable, it's more reproducible, and there are fewer regulatory agencies. So we take cotton and we do the healing technique. This goes back to the great Bernie Grad, who discovered that healing can be stored in cotton. It seems to turn into a universal donor. And the question is, if we juice up cotton, can life recognize it and can gizmos recognize it? So we can store the healing in the cotton. Can the mouse recognize it? Can a gizmo? Do you have anything that can recognize whether it's been stored? And does it make a difference if there's a healing need? Now, healing is a response to need, I think. Here's what happens. In my particular healing technique, a couple of odd things happen. One, everybody becomes left-handed. Curious, but it's universal. Everybody becomes left-handed, and the mice, if you can see there, will try to go to actually this exact spot on the left hand. We spin the cage, they'll go to that spot on the left hand. When they're cured, they no longer go to that spot on the left hand. It makes no difference what kind of mouse it is. Once they're cured, if we had this in a motion picture, you'd see them rotating to get to the spot on the left hand. Once they're cured, they don't do that because they have no need. So, we juice up some cotton, and then we put cancer cells near it. So we have cotton that's charged, we have cotton that hasn't been charged. If we have cotton that hasn't been charged, and we put cancer cells near them, nothing happens. If you put cotton that's charged, you put cancer cells near them, we get genomic changes. Cancer cells can recognize whether the cotton has been charged. Can gizmos, technical term, detect charged cotton? So we've done all sorts of stuff with all sorts of fancy gizmos, and people are talking about various, I don't know what the plural is, maybe gizmi, various gizmi, and what can they do and what do they see, and we can't find a gizmo that can tell whether cotton has been charged or not. I've even put it under an electron microscope. I don't know what I was looking for, but can you tell the difference between a charged piece of cotton and an uncharged piece of cotton? We can't find anything to date that can do it except life in need. A mouse in need, a cell in need, something that's wrong, they can recognize whether cotton has been charged. This is me playing Men in Black. With Dean, we were doing some infrared stuff. And here's the question. Can we record the charging of the cotton, and then can we play it back and reproduce the healing? Can we take the humans out of the equation, thereby making it scalable? So, this is Faraday cage at ions. It's a big sucker. It's heavy sucker. It's a spacious sucker. If you've never been in a Faraday cage, you want to do it. It's a lot of fun. And we put stuff in there, 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 we put more stuff in there, we put more stuff in there. We put 38 detectors in there. And the reason for the 38 detectors is simple. We have no idea what we're doing. And so what we're trying to do is maximize the possibilities. So if we can go from 1/100th of a hertz up to, can we capture anything in there that'll be useful? That's the question. So not knowing and having no theoretical basis, we're throwing stuff at the wall and seeing if anything sticks. That's the general idea. So we got 38 detectors in there. We had to build a water process supercomputer because we got a monstrous data set, and we did a whole bunch of iterations of things inside. The most significant thing that we did inside the tube is we had 3 people using my healing technique holding pieces of cotton. That's all they were doing. They're holding pieces of cotton, and 38 detectors are running inside this Faraday cage. What happens? What is that signal? And will the replaying of that signal reproduce the phenomenon? So we've looked at objective measures, we've looked at all the gizmos, we looked at all this kind of thing, and we can't see anything that's particularly significant. Mike Turner did a whole bunch of the signal analysis. There's nothing there that would account for functional curing. So there's little weak chirps, and there's little this at very low frequency and little that, but that's not going to account for the phenomenon of healing happening from here to Australia. So let's be real. The kind of little chirps that we're getting, it's not explaining the phenomenon. This is what I mean by we don't know what we're doing. So what happens if we make a recording and we play this to cancer cells? Will they be able to recognize it in the way that they were able to recognize the charged cotton? So we get incubators and we stick cancer cells inside the incubators and we have speakers. Now I got to tell you this, just for perspective of scale. It cost us a little over $300,000 to make this recording. We bought an amplifier for $49 from Amazon. And I think we got expensive speakers for $50. So our hardware there on that end was slightly under $100. And so we do this, and we play, we put cancer cells in there, and we play this recording to them at various dose responses. And we look at it at 4 hours and 24 hours and 48 hours, and we look at cancer cell, we look at the genes, 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 we look at a lot of genes. We've looked at 167 genes. A couple of them seem promising. They seem to have to do with inflammation, apoptosis, telomere length. For this part, we have 68 out of 167 producing reasonably reliable genomic changes. The recording and the signal analysis, which I'm skipping here for time, there's almost nothing in it. Yet this thing that has almost nothing in it is producing biological effects. Cancer cells can recognize it. So 68 out of 167 have significant gene changes. We can give as a tentative conclusion that intention can be captured and released. Maybe we can scale it. We've tested the recording versus people doing hands-on. And it turns out the recording is we think the recording is a little bit weaker than hands-on. We don't know why that is, because we don't know what we're recording. But the hands-on healing has not been completely captured, we think. We don't know if that's because of the digitization, and we've lost information by digitizing. We got to replicate this by analog, et cetera, et cetera. But hands-on is a little bit stronger than the recording, at least in terms of the genomic outcome. These first three, incidentally, are now going to be published, I hope, in the next week in a conventional biology journal. So this is reasonably significant. This is not an alternative thing. This is a conventional biology journal that said, whoa, yeah, let's do this. We left out the fourth conclusion. Conditioned space. If you play the recording to people who are sensitive, they go, ooh, ah, and they flop on the floor, and they do all those kinds of things. And then when you turn it off, this is 100% of the time, the sensitives say, it's not off. I hold up the plug and I go, no, it's unplugged, it's off, it's unplugged, all that kind of stuff. And I go, it's not off. So sensitives are annoying, but you got to follow their lead. So what we tried is we played the recording, and then it goes in, and you get the genomic changes in the incubator, and then we turn off the recording, and then 3 days later, we put in more cancer cells. Damn sensitives. They still change. We've conditioned the space. Now this would have incredible methodological implications, another talk. If you're doing an experiment and space can be conditioned, see where I'm going? This is reasonably interesting. Next steps, which I'm not going to get to very many, we're doing now replicating by playing the recording to mice and doing further signal processing and all that. And so we've just finished. We don't have the data in yet. The talk would be different 2 weeks from now, as it would be different a month from then. We injected cancer cells into the animals, and we tried different doses of the recording. Here's a quick picture of the setup in Providence. Same idea. We've got the speakers, we've got the amplifier, but now instead of cancer cells in an incubator, we've got mice in a mouse room. You can see we got ulcerations again. We had a complication here. I got to stop. I'll talk to you on the side. Something's happening to the mice, folks. It's reasonably interesting. We have much more data to come. Stay tuned. Thank you to our funders, and thank you.
Open for question.
Yeah, I'm trying to figure out a possible contradiction. In one of your early slides, you said intention didn't matter, and then in one of your conclusions, you said that it did matter. So
no,
which is it?
I think I either misspoke or you misheard or something happened. Intention matters, attention does not.
Attention.
Attention does not.
So state of mind would matter because it's a state of joy, apparently, as you're cycling through the...
Nah, there's no joy. The most dominant thing you'll feel while healing is just utter God-forsaken boredom.
Okay. If that works, I got plenty of that.
Yeah, it's boring. To do this hour after hour is exhausting.
So, Bill, you mentioned telomere length. Can you tell me, were you getting longer telomeres as a result of your healing?
We think. Too early to conclude for sure.
But the answer is yes, you think so?
We hope.
But did you think that's what you found?
You're not going to trick me. It's not going to happen.
So the mice are attracted to a certain point on your left hand when they need healing. What about people? What do they feel?
Same thing.
So do they feel?
Most adults will talk themselves out of a natural flow of anything. So if you take a mouse, it'll go to the left hand. If a little kid will do the same thing. So if a little kid, you don't argue with a little kid, you got a 2-year-old walking around, the 2-year-old will walk to that part until they're cured.
What do they say? What do they feel?
Like they should be there.
Thank you.
Bill, you mentioned that these results, or at least your first 3 conclusion points, are going to be published in a mainstream biological journal within a week.
We hope.
What journal?
Dose response. Yeah, once it's out, I'd be happy to ship it to anybody who wants it.
So the purpose of a cancer cell is to grow out of control. It has no regulations. So the cell lines that you're doing when they're given the healing, they just do what they're destined to do. They grow exponentially. Are there cell lines that aren't cancer cell lines? And have you tried any of this on them?
In the it's just ordinary cancer cells?
No. Ordinary cells, not cancers. Are there?
I did that once at I'm trying to remember which university. I've been in too many labs. Nothing happens. Without a need, nothing happens.
Okay, the normal cells don't grow out of control because that's not what they're programmed to do.
No.
Okay.
My guess is then you're trying something like a PK experiment. And that would probably inquire not only intention but attention. Healing does not conform to your wishes, desires, direction, plans, chapters, however you call it. Healing happens on its own terms, and we're trying to understand the terms.
Thanks so much for the talk. I have someone in my family who recently came down with cancer, and they're reasonably open-minded. Not as much as all of us, but reasonably. So do you need any more guinea pigs?
Well, we don't use clinical situations as guinea pigs. There are people who practice this technique all over the place, and we could talk.
Okay.
But I don't do experimentation on humans. The reason I got into the lab is after we had a few hundred of human cases, I wasn't learning anything, and I couldn't tell why something was happening. So in the case of a clinical condition, did they get better because they ate a grapefruit or because they didn't eat a grapefruit? In the case of lab work, I can control the variables much more.
All right, thanks so much. I'll be in touch.
Yep.
Great talk, as usual. Can you say as much or as little as you want to now about how do you know when healing is being done? In terms of what the humans are doing?
Oh, very simple. I have no idea, and it's a complete surprise to me. There's not going to be a single pattern. We tend to focus on central tendency. Here, what is the aggregate or what's the average or something along those lines. But on any individual case, there's an enormous unexplained variance. So I can't tell when something happens, but what happens in mice, in men, in John Steinbeck, no matter how we do this, you never know when John Steinbeck is going to make that phase transition. But it almost always occurs that way.
You have humans involved in the experiments, right?
Only as volunteer healers. Yeah.
But we talk about healing. I mean, if you just had the mice, I remember you talked about this at length in previous presentations, and it looks like we're running out of time.
Morris is going to say, let's talk at the back of the room.
I think so. Thank you very much for a great talk.